Welcome to the ArthroPod, the podcast where invertebrates aren't just weird, they're wonderful. I'm Dan, your host and lifelong bug nerd behind Planet Critters, and in each episode we'll dig into the science, care and curiosity behind exotic pets, entomology and the tiny ecosystems that often go unnoticed. Whether you're here for the beetles, the biology, or just to geek out about things with too many legs, you're in the right place. Let's get into it. So, I think we will start with the question, why did I start the podcast? I think mainly I must just like the sound of my own voice, because I keep putting myself in these positions where I can make people listen to me. But also, I was looking for a podcast about entomology and zoology, and there were very few that hit the mark for me. There is some good ones that I listen to. So Tom Moran's podcast is awesome. There's another one I like that's more zoology based called Tooth and Claw, but there's not a lot out there, to be honest. So I thought I'd see if I can make one myself. One that covers both the hobby of keeping invertebrates and exotic pets and looks at the science behind entomology. After that, the next question was, what do I want from the podcast? First, I wanted a space to explore my own thoughts, feelings, and ideas in this hobby and profession. Next, I wanted to be able to develop my scientific communication skills, hopefully providing some educational value to you folks listening, and maybe even give a little bit of entertainment value, or at the very least, some background noise for you while you feed all your pets. So I guess our very first segment is actually going to be a bit of a self-indulgent one for me, because it's going to be an intro to who I am and how I got into this hobby. So my interest in the natural world and arthropods started years ago, back when I was a child. I came from quite a poor area. So for those of you who have heard of the Scottish Index of Multiple Deprivation, I live in an SIMD2 area, which is the bottom 20% of earners in the country. So it's an area of abject poverty in Scotland. We didn't have a lot around us, but what we did have was some wild spaces. So there was a couple of fields around, and I had a fairly overgrown back garden in a council estate. I was always finding wee beasties and critters and putting them in old Chinese takeaway tubs and looking after them for a while. I made a couple of ponds in my garden by just digging a hole and filling it with water, which didn't last very long. But I was always encouraged to do this. So one thing I had that a lot of my friends around me didn't have was a very nurturing family. So I had a family that always pushed me to do what I wanted to do and really encouraged my interests. I also had a massive idol who really pushed me along this path of loving animals, and that was Steve Irwin. So for those of you guys that grew up in the 90s and 2000s, you will probably know who Steve Irwin is. He is the crocodile hunter, and he is a massive inspiration for animal conservation and zoology worldwide. The man was amazing. When he passed, I was absolutely distraught. I cried. It was like a family member had left us. I was so upset. But it didn't put me off wanting to work with animals one day. I was adamant that I wanted to be a zoologist, but at the time, I didn't necessarily know what that meant or what a zoologist was. When I went to school, I wasn't very interested in school. I wasn't all that interested in education. I didn't leave with very many qualifications. I've got one in art. I might have geography. I didn't even go to my maths or English exams at the end of the year. Yeah, I kept quite a lot of school, so I truanted quite a lot of school. And I was actually kicked out of science at some point early in school, which we'll come back to later as to why that's a little bit ironic. But when I left school, I was lucky enough to get involved in outdoor education. So I started teaching things like kayaking and windsurfing and sailing and mountaineering and rock climbing, as well as other aspects of outdoor education, like the John Muir Award and Duke of Edinburgh and stuff. With a lot of the qualifications that I got from those hobbies, I earned UCAS points. So anything that is what we call a national governing body, so it's an organisation essentially that looks after something or over a sport. If you complete a qualification within that NGB, you will get UCAS points, even if it's just 5 or 10. And with a few of those, I had enough UCAS points to apply to a foundation degree at university. Something I never really thought I would do was go to university to study science. But my girlfriend at the time had moved to Aberystwyth to study science, and I just kind of followed her. She studied for a year, then I applied the year after, realising that I could. I managed to get on a foundation degree with or zoology with a foundation degree. So I done a year's foundation, and then I moved into a Bachelor's of Science with honours in zoology. And I was able to complete that year. My dissertation was looking at mate choice in a type of isopod, a brackish water isopod that's quite common in the UK. So looking at whether they display preference for different sized males. So it was female mate choice as well, which hadn't been discovered. And I kind of, like my research pointed to the fact that they do, but there's a few other things we needed to improve on to be able to say that for certain. While on that undergraduate course, my bachelor's, I was part of a scientific expedition that went out to the Peruvian Amazon, to primary rainforest in the heart of Peru. Now, when we landed in Peru, we then had to get another plane for a couple of hours out to a more separate area within Peru. I think it was Maldonado. And then from there, we had to get a minibus for about 3 hours to a little village on the banks of the, I can't remember the name of the river, but it was one of the bigger rivers in the Amazon. It wasn't the Amazon, but it was one of the bigger rivers. I think it was called the Devil's Mother or something when it was translated. But I'll need to look that up. But yeah, it took us about 3 hours to get to that river. Then once we were at the river, it took us about 7 hours on the boat to get to the research station at Los Amigos, where we were based at. So quite a long journey to get into this part of the rainforest. It was one of those areas we had to be very careful when we were out there, because if something went wrong, it was a long trip back to the hospital. But it was an amazing time. I was working on the Barcode of Life project, so we were sampling a whole bunch of inverts. We were taking genetic samples from them, storing it, bringing them back to our lab in Aberystwyth, and then processing them to add them to the Barcode of Life, to a giant database where we try and essentially have a genetic barcode for every animal that exists. We're never really going to get there, because it's impossible to know everything that's alive in the world with the way speciation occurs in some of these areas and the way that things are dying out before we even get a chance to sample them. It's impossible. But it's a really cool project, and anything we can do to further scientific knowledge is quite cool. While I was over there, I was lucky enough to meet a man called Dr. Ross Piper. So he is an entomologist, and if any of you recognise the name, that is probably from the show Wild Burma, which was on the BBC, where he was the lead scientist and host in an expedition into Burma, which had previously never been studied by scientists. It was a completely wild and remote area. While there, they found an incredible diversity of animals, a whole bunch of new species, and a new genus of mantis. I believe it was mantis fly or crane fly. I think it was mantis fly, which was a really cool discovery. And they're still writing papers on that, on some of the diversity that's coming out of there. It's amazing. It's something I would love to do as well at some point. But yeah, I met Ross, and he absolutely got me enthralled in entomology. Before that, I was thinking that I was going to be a herpetologist. So I was looking at reptiles and amphibians and stuff, which I still absolutely love. I've got a couple of snakes and a few amphibians myself. My favourite animal is probably still an axolotl, which are really cool. I've got a couple in my collection. But yeah, I fell for entomology. And from there forward, that was just what I wanted to do. I was lucky enough when I was over there that I have perhaps discovered a couple new species of insect. So we're still waiting on some of the work being done for that. We've done a lot of the genetic analysis, and we are pretty sure that they are distinct species. They are currently sitting in the Natural Museum of History in London, waiting for an expert in morphology to come along and just confirm our findings as these being new species. But we've looked at a whole bunch of dichotomous keys, and we can't find anything that is this animal. And as we say, we've done the genetic work. It's never been barcoded before, it's never been sampled before, so we're pretty sure it's a new species. But that's really cool. We're waiting for that to come back. And then when it is, I will eventually write a paper on it and hopefully be part of a description of it. If, at very least, I will be credited as the person who found the animal. After that, I came back with this new love for entomology, and I decided I wanted to do my master's as well. So I went into a master's of research with Dr. Joe Ironside at Aberystwyth University, and it was looking at fungal parasites within freshwater ecosystems, specifically looking at fungal parasites that infect a group of animals called the amphipods, which are a type of crustacean. Quite often they get called water fleas, sometimes killer shrimp, stuff like that. There's a few common names, but the amphipods they're a really cool group of animals, and I was looking at freshwater ones. The fungal parasites that I was looking at were what we call microsporidia. They're really cool. We used to think they were animals. We now believe they're funguses, although it's not 100% on that. And one of the things these animals or funguses can do when they infect their host is they can feminize them. So they exist, or a lot of them exist in the ovaries of animals, and they infect the eggs. And then when they're in the eggs, they will sway it towards developing into a female of that species because they can only exist in the ovaries, really. Problem with that is, if you turn all of the hosts that you encounter into females, and it's not a species that can reproduce asexually, so it still needs a swap of gametes, it needs a male and a female, then you're going to die out within a couple of generations because your hosts are going to disappear. So there's a balance between making enough females that they can really proliferate, but not so much that they lose males and the hosts start to diminish in the ecosystems, which has some really cool implications for evolutionary biology and phylogeny, which is what we were kind of looking at. I was able to get together a whole bunch of funding for this project. So I was working with the Centre of European Fisheries and Aquaculture Science. I had use of a lab in London with them. I had a couple of people that were going to be working on my project where I could send them some samples and they would be looking at it. I was doing an integrative approach, looking at genetic identification as well as morphological identification using something called histopathology, where we would essentially be cutting up some specimens into really, really thin slices and then examining them under the microscope, looking for physiological characteristics of disease. So stuff we could see in it, physical things. But this was happening in the 2019-2020 period. So if you remember around then, there was a few things that happened. The first one that really affected my research was Brexit. So the UK decided to withdraw from the European Union. It's actually a bit controversial to say the UK because the country that I'm from voted against it, but they were still withdrawn. So there's still arguments over that up here, unfortunately. But with that, a lot of the funding for science came from the European Union, and that was withdrawn, which I guess is quite rightly so. If you're providing it to members of the European Union, if there is a country that isn't part of the European Union, then arguably, why are you providing them funding? So yeah, that funding was withdrawn. The UK government had promised to redistribute more funding to a lot of these research institutions that had lost the funding, but it never really happened. I was lucky enough to get a little bit more funding together to be able to work on some of this stuff. So again, I got some more funding, not so much in money. This time I was more allocated hours at the place that I'd originally had the funding for. So they had given me some hours of staff there and some lab hours that I was able to use. I was getting ready to go down for my first visit to the lab. So this was the Monday I got everything cemented. I was planning to travel down on the Wednesday and then kind of work through Wednesday afternoon, evening, and Thursday, Friday, and then come back up on the weekend, back up to Aberystwyth. As I was packing, I got a notification from the university, and that was that the labs were all shut down. So I kind of knew it was happening because there had been a couple of things happening on the news. And from that period, you'll probably know what it is straight away, and it was COVID. COVID had started to really become a problem in the country. It was spreading through the UK quite severely, and they had restricted travel, but they had also closed down all the labs at Aberystwyth University. The lab I was working in was actually turned into a COVID test processing centre, essentially. So they took all the labs that had the equipment to sample genetic samples and turned it into one for looking at PCR tests and stuff to identify whether people were positive for COVID. So yeah, my lab was shut down, lost all the funding again because the labs in London were also shut down. And with something like 3 months' notice, I had to change my entire project, a research-based project where I had about half of the data that I needed to be able to finish it, into a lab-based project. So I started to put together a massive database of all the freshwater, and I even started to move into marine invertebrates that we know are associated with these fungal parasites and of the fungal parasites themselves, and look at linkages across them, possible zoonosis for them transferring from other animals into humans, which is something we think can happen. In fact, these parasites might be involved in human immunodeficiency virus, so HIV. And yeah, it wasn't a very fun time for me. I went from doing something that I really wanted to do, so research, into something that was a bit more boring, just writing stuff on the computer. But I got it finished and I left with my Masters of Research in Bioscience. So I got my undergraduate Bachelor's of Science with honours in Zoology and my Master of Research in Bioscience. So there's three main kind of Masters postgraduate courses you can do. You can do a Masters of Science, Master of Research, what I've done, and an MPhil, which is really designed to move into a PhD project. I am actually playing around with the idea of going and finishing my PhD, but it is highly competitive in the UK now, especially once that funding from the EU was removed, and it's never been put back since. And we are what now 5-6 years down the line. It's quite competitive. So I'm looking at perhaps putting together my own PhD proposal. But there's also a few other things I'm looking to do. So I'm looking to move back into academia as well. I'm looking to do it as a freelance scientist this time though. So I've got a couple of scientific papers in the pipeline I'm looking at doing. One was based on some of that work that I was doing in Peru. So while I was there, I was looking at Calafia lutea, which is a large flowering plant, has these massive leaf rows that shoot up out of it as the new leaves are developing. They're all rolled up as they come out of the stem. And within that, you will find quite a big diversity of invertebrates, quite a lot of beetles and stuff. And when I was over there, I'd done a little project where we were cutting off these leaf rolls, we're unraveling them and sampling all of the inverts that we could find in it. And we were putting them into distinct morpho categories, so animals that looked roughly the same. So not quite the genetic work that I was used to, but looking at morphological characteristics and separating them like that. And then we were starting to look at, or really hypothesizing some sort of theory for island evolution within these leaf rolls. Should be really cool. So I've still got a lot of the data for that. I'm going to see if I can bring it all together and start to write a paper. We were supposed to be doing this with a researcher in America who got a lot of our data and was supposed to be getting more, but I've just never heard back from them since. So I'm assuming that that's not happening anymore, and I'm going to look to take what I've got and publish it myself. And then I would actually like to do something similar in the UK, but I've not decided what I'm going to look at yet. There's a few things I got for that. I also want to do a paper looking at the diversity of saprophilic animals in fragmented habitats in the UK. So fragmentation is when habitats are split up and they're kind of divided, maybe by roads or just fields or breaks in forest. And saprophilic animals are animals that's lifestyles revolve around dead and rotten wood. So it's a really endangered habitat in the UK. As forests decrease and as forests themselves become a rarer habitat, these deadwood habitats become much rarer at a higher, almost an exponential rate, really. And there's a few other cultural things that kind of feed into that. So one of the things I've started doing in my local area is I have taken on a uncared for little piece of woodland behind my house. I have no idea who owns it. The council doesn't seem to know if it's them. Nobody local seems to know who owns it. I can't find any deeds or anything to say who owns it. It's just almost like an unowned piece of land. So I've started doing some work in there looking at conservation. There was a big beech tree that was blown over in a recent storm. So I've got a little electric chainsaw, geared up with it, threw all the batteries in the bag, went and spent about 3 hours clearing this tree out that had fallen over the path where I walk my dog. I decided to clear the path, get it out of the way. And I stacked up some wood and then scattered a few of these logs around the forest, thinking this will be brilliant habitat in a few, over like the next year or so. It will start to break down as the different fungus in the area get at it and start to rot it. And I went back the next week and somebody had been in and pinched it all. This is one of the problems we face in the UK as wood becomes more valuable, as wood stoves are starting to come back into fashion, as wood-fed hot tubs are coming into fashion, as wood furniture is more expensive, as just lumber in general and timber is more expensive. People are going into forests and taking this wood, and it's never getting to the point where it is becoming deadwood. So these habitats I was really excited about being able to survey in the future were just entirely gone. But I'm going to try and survey some of these. There is some deadwood there in an area that people don't really go to that looks like it could be prime habitat, which I'm going to likely survey next spring. So you never know, that might even become a paper in itself, which would be really cool. But I'm going to do a massive literature review looking at a lot of these habitats across the UK and how diversity is increasing or reducing. Very likely reducing. The last one is I'm starting to outline a paper in the tarantula space. So I'm going to be looking at likely the adaptive morphology within the genus Arcanthuscuria. So it's a genus I really love. I really love the Acanthouscuria tarantulas. I've got a pair of Acanthouscuria sternalis, a breeding pair behind me, that are going to be a project in the future. Neither of them are quite mature yet. I did notice on the last molt that the spermatheca of the female is slightly scleroterized, which means basically it's starting to get a little bit harder, which means she's getting to the point that she's able to breed, although she's still a sub-adult. She's getting to the point that she is mature. And the male doesn't have the mature reproductive organs yet, which are those emboli that we see on the end of the pedipalps, little hooks. So I'm just waiting for that to happen, and that'll be a project. And I've got a little sling Acanthouscuria geniculata, which is the Brazilian white knee, which will be really cool to look at as well. But I'd love to be able to look at some of these animals in the wild when I'm looking at this adaptive morphology. If not, it'll be largely literature review as well. So a couple of little things in the pipeline. More recently, I got kind of distracted by my academic journey there. More recently, I have moved back to Scotland. So I completed my masters. I got my masters in bioscience. Then me and my partner decided we'd kind of had enough of Wales. It's a cool place, but our hearts are in Scotland. We love Scotland. A lot of people that live in Scotland, we have a huge bias for the area. It's amazing. I'm not sure I could live anywhere else. We moved back to Scotland. My girlfriend is also has a science background. She also done her BSc in zoology at Aberystwyth. She also got her master's in bioscience. She was looking at genetics as well, although she was looking at much harder genetics than I was. She was developing new novel barcodes for identifying flowering plants and then mosses in her master's, which is something that's still being worked on. She was really at the forefront of science for that. Worked with an amazing guy called Professor Mike Wilkinson in Aberystwyth, who was a really, really good lecturer and professor. And an amazing scientist, like world renowned for some of his genetic work. So yeah, she came back to Scotland and was able to get a job as a geneticist for the Scottish Government, where she worked looking at Scottish raspberries, which was really cool. So she was looking again at novel barcodes for identifying Scottish raspberries. And she also worked with a little bit of commercial science, identifying potatoes. Yeah. One of the things was she was working on this potato that was set to be the world's biggest potato ever grown. It was this farmer in New Zealand, and he had sent over a sample because at Sasa, where she worked, they were famous for working with potatoes and identifying potatoes from all over the world, telling them how healthy the crops were and stuff. He sent this sample from his biggest potato to confirm that's what it was, and he wasn't very happy when it returned. It was actually a larger tuber of a different plant rather than one of the potatoes. He really wasn't very happy with that, and it kind of dashed his world record. But that's what science is. Sometimes it doesn't tell you what you want to hear. So yeah, she had some really cool projects that she was working on over there. Fortunately, again, she decided to leave, and that is not because of the science, it was because of a colleague she worked with. Her manager was basically a bit of a bully. Those of you aren't aware, the Scottish Government has had a little bit of controversiality recently for nurturing an ethos of bullying within many of their kind of governmental groups, and she really noticed that when she was there, which is unfortunate. She has then actually been reached out to by someone who still works there, who is having the same problem with this manager who is still there. And I think she's basically told them the steps she went through, and that's going to hopefully move to a formal thing. But they need to get rid of these people out of science. They do nothing for science. They're not particularly good scientists themselves a lot of the time, and they just put people off, because she was actually a really good scientist, really clever. But yeah, she worked in science. I didn't. I was a bit sick of academia after everything that happened with the funding, and I decided I wanted to take a slightly different career path for a bit. I just got a job for a while working in a garden centre, saved up a little bit of money, and then I went and completed my PGD. Well, I say I saved up money. I spent a lot of my money on plants and stuff, but I saved up a little bit of money, and I went and completed my PGD. So that is my postgraduate diploma in education. So I am now a fully registered teacher of biology in secondary education. So I can teach biology and science at secondary schools. I am also working on getting registered as a fully registered teacher for primary, which would be really cool, but there's a few things I need to do for that. So teaching has always been something that I've been naturally drawn to. I don't know what it is. It's never something I particularly planned on doing. It was just kind of something that I kept falling into, and it's something I really enjoy now. I would love to produce a couple of papers in education. So one of the things I'm really interested in is a holistic approach to education. Holism has got this kind of bad press as being unscientific. When you look at the science of holism, it's the way that all the parts integrate to make something larger than itself when combined together, or how each part can be more important than kind of the end result. It's a really cool thing in science, and when you integrate into scientific systems, it can really change how you look at certain things and how you teach certain things. So I was wanting to look at a holistic approach in science where we think about what's important in what we teach science. So I'm a big proponent for a lot of the things you learn at school. The academic lessons are not the most important. The most important are the lessons that progress self and emotions. Okay, which is a weird thing to say in science because we think of science as being very cold and very extracted from stuff like that. But I think if you want to make scientists, you don't need people who can tell you what the reactive products of enzymes are or how enzyme specificity works, which is something we have to teach and it is entirely boring. I think what you need is people to be interested in science. You want to show them some of the cool animals in the world, which for biology is a lot of the reason people study it. You want to say, this is what you could be working with. Or you want to look at poisons and toxins and say these are the types of things you could be working with. You could be curing cancer one day rather than the boring shit that we teach them. So I'd like to produce a paper showing that and showing that that's what we need to focus on rather than these academic outputs. Get away from stupid tests that mean nothing and actually teach people what they want to teach and what they take science for. So I've completed that. I am working as a biology teacher at the moment. It's very few and far between the jobs, so I'm looking at a few other things. I also work with the Royal Zoological Society for Scotland as a discovery and learning associate at Edinburgh Zoo. So I deliver conservation action education programs, working with people to teach them about the conservation work we're doing at Edinburgh Zoo, some of the outreach conservation work we're doing, which as a charity registered zoo you're required to do, and also working with school groups and stuff that come into there, delivering some other educational programs. It's an amazing thing that I love doing, although I don't do it all that often, just a couple of times a month. I do it on my weekends, and it is absolutely incredible. I am now playing with the idea of getting back into academia. So I mentioned I was looking at PhDs and stuff. I'm looking at pursuing a PhD, perhaps in entomology. I have also inquired about a few in education instead, which would slightly change up my career route. But it's hard to decide. I was the first person in my family to finish really secondary education, to finish high school, never mind going on to university. I've not really got many people to ask, so I'm just kind of trying to stumble through this as I go. I am also, as I said, looking to publish. I'm looking to publish as a freelance entomologist rather than working through universities or other partners, and I'm kind of held to their whims. I'd like to try and do it myself. A lot of the surveying and stuff, if I've got time, I am able to do this survey, and it doesn't necessarily cost a lot. And I can perhaps look at publishing open source, where people can just see my work and I pay for the publication rather than it being a journal based system where you have to pay to see the paper and that's how they make their money. Although I will still probably apply to some of these journals that have reading fees as well, just to get some of my work out there. So hopefully, after that 20 odd minutes of me telling you who I am, you have a little bit more insight into who I am and a bit of an idea about my academic background. So my entrance into the hobby wasn't really a hobby entrance. I didn't start with the pets. I started really with the academic background. My mum hates snakes. She's told me she's never visiting while I've got a snake. And my sister hates spiders. I only recently got her to visit the house. So it was never something I could have growing up. And it is only recently, after completing all my degrees, that I have moved into keeping these animals that I was lucky enough to be able to get to see in the wild on a few occasions. In Florida, in Australia, in Hong Kong, and in the Peruvian Amazon. So yeah, this is just me kind of starting to catalogue my journey through this hobby and give you some of the academic insight that comes along with it, which I think gives me some slightly different ideas about the hobby compared to other people who are in it from a purely pet keeping or a keeper's perspective. I'm slightly outside of that in some of my thinking, which I think could be quite valuable to have a few more voices in the hobby. Now, let's get into some science. So firstly, do you guys know how many new species of Theraphosid have been described this year? So Theraphosids are the group that we commonly refer to as the tarantulas, or the large hairy spiders. In fact, before we step into that, it's maybe worth taking a little step back and answering the question, really, at the heart of all this. What is a tarantula? Because they're not just big spiders, they're biological marvels. I've got this bit of almost a chip on my shoulder about this term that a lot of people have been using recently, the true spiders. So we're in the literature, we're trying to move away from the true spiders, and we're trying to talk about more the advanced spiders and the primitive spiders. The reason they get some other spiders, the Aranidae, get called the true spiders is because they're what we call the spider form spiders, whereas the tarantulas are not considered spider form spiders, which is just silly. So we tend to talk about the primitive spiders and the advanced spiders. Now, as I said, these tarantulas belong to the family Pherophosidae, and it's one of the largest and most recognisable families within the class Arachnida. So they are part of the order Arani, which is all spiders, and within that they're part of the suborder Megalomorphae. So you might have heard of the megalomorphs versus the aranids. And this includes other primitive spiders as well, though, like trapdoor spiders and funnel webs. These megalomorphs are often considered more ancient in terms of evolutionary development. Unlike modern spiders in the aranomorph groups, they have vertically moving fangs. It's kind of up and down like that. They're a bit like kind of two spikes or spears stabbing down, rather than the pincer style fangs you see for a lot of the aranomorph spiders, where they're almost coming across themselves like that to help grab prey and hold on to it. You can see them quite clearly in the orb weavers and the jumping spiders, some of those bigger ones. The big huntsmans as well, they're really easy to see them crossing across like that. We say there's currently about 1,000 recognised tarantula species across the globe, and they're distributed right across the Americas, through Africa, Asia, and some of the southern parts of Europe as well. It's a little bit warmer. They're most diverse, like most things, they're most diverse in South and Central America, with hotspots really in the Amazon rainforest and some of Mexico's dry forests, which are pretty cool. But you'll also find them in places like the deserts of Namibia and the highlands of India and caves of Southeast Asia. There's a lot of cave tarantulas coming out at the moment, especially out of Thailand. These spiders have adapted to a huge range of habitats. We've got arboreal spiders, which are the ones you're going to see tree dwelling, some even right up in the canopy, about as far up as you can get in the trees. And then we've got some of these fossorial spiders as well, whereas these spiders that spend a vast majority of their lifestyle burrowing and underground. Or terrestrial ones, which spend a lot of time on the forest floor, moving around, and these tend to be at least semi-burrowing species as well. Some live in places like humid cloud forests, others in harsh scrublands. But what kind of unites them is what we'd call a rugged physiology, which you can kind of see if you look at them, and this slower, more patient approach to life than you might see in some of the jumping spiders, some of the orb weavers and things. So tarantulas are ambush predators. They tend not to spin webs to catch prey, although, again, controversial because it depends on what you consider spinning a web, is leaving a trap wire to recognise when prey is moving about, considered spinning a web. It's a little bit hard to say in the literature, but we tend to say they don't spin webs to catch prey like your stereotypical spiders, which are your araniomorph or your spider form spiders. Instead, they're going to rely on stuff like stealth, strength, just overpowering prey, and venom to be able to subdue their meals. Usually insects or other arthropods, but also small reptiles and occasionally amphibians. You might think as well, if you've heard of the bird eaters, that they'll prey on a lot of birds. It's not really something that has been documented. We definitely have surveyed spiders that have been eating birds, but it's likely they were scavenged rather than actively hunted at the time. So if you see bird eaters on birds, it's probably birds they've found, not birds they've caught. It's still unclear as to whether they actively hunt birds. Very unlikely, though. Birds are not an easy prey source to take down. The fangs in some of these megalomorphs, they tend to be quite larger, are up to 2 centimeters long in the largest species. We have got a T. apophysis behind us, which is one of the biggest tarantulas in the world. And you're talking about, when that thing's older, the fangs are going to be like that length. They'd be able to pierce my finger and show up the other side quite easily. So quite big spiders. And they tend to inject venom that will both has enzymes to paralyze and start to digest the prey. And depending on where they come from, it tends to be stronger or weaker. So we tend to say old worlds usually have a little bit stronger venom, and new worlds have slightly weaker venom. And that's probably to do with the defensive properties of the tarantulas, and the new worlds are really the tarantulas that have those urticating hairs we talk about, which are the little irritative hairs with barbs on them that can produce nematocysts in them as well to actually cause harm to people. So, I mean, when we're talking about the venom, though, although people are kind of scared of tarantula bites, the venom's typically fairly mild for humans. Most of them not that much worse than a bee sting, really. It's the mechanical damage from the fangs that's more of a concern than how toxic the venom it is. So if you get hit by a ferrophosit, like a big one, it can do some damage to your finger. It can go in quite far. And the problem with that is when you've got a wild animal, there is going to be bacteria on those fangs. And then once infection sits in, that's where you see most of the problems coming from, not the venom itself. The venom, even some of the most venomous tarantulas, the ones that we might consider medically significant, it's not going to make you feel too much worse than just a bad hangover, when you're poisoning yourself anyway with these toxins. So yeah, normally the size of the fangs is a bigger concern than those fangs, than the venom. I mentioned the urticating hairs, so that's one of the defining features, really, New World tarantulas, those for Americas. And it's their use of urticating hairs. These are tiny barbed bristles found in the abdomen, which they can kind of kick and flick at predators using their hind legs. These hairs can cause quite a lot of irritation to skin, and especially eyes and other sensitive areas in mammals. But they are a really cool example of non-venomous defense, and quite specifically probably for mammalian threats. So the venom's kind of really been adapted for use on insects and small mammals, whereas the hairs seem to be aimed at defending them from large mammalian threats, which is really cool. Old world tarantulas tend not to have these hairs. That's perhaps why they've got a slightly higher toxicity in their venom, and they tend to be quite fast as well, relying on that to get away from predators rather than warding them off. Tarantulas, as well, they tend not to see as well as some of the other spiders. They tend to have around 8 eyes, but their vision is quite poor. Instead, they rely a lot of the time on vibrations transmitted through the ground and the air, detected using fine sensory hairs across their legs and body. So their entire world is kind of picked up on tremors and air currents and ground shifts. A bit like Daredevil. So these are like the little daredevils of the rainforest or the desert, which are really cool. They grow by molting, so they shed their exoskeleton to allow for growth. Some species can live 15 to 30 years, even longer, I think, has been recorded in some of the grammostolas, but that's mostly with the females. Males tend to live maybe 5-6 years. Although there was confirmed, I want to say Laceadora parabiana, that was about 12 years old, male, but males tend not to, they won't molt once they get to their ultimate shed or their ultimate molt. They won't molt again. There has been some examples of males molting once mature, but they tend to die shortly after the molt because those pedipalps don't molt very well, those hook-like structures in the pedipalps that develop in mature males. And yeah, so the next time you see a spider, don't think, oh, that's not a true spider. I mean, next time you see a tarantula, don't think that's not a true spider. It's absolutely a true spider, but it's one of what we might call the primitive form spiders rather than the advanced or spider form spiders. But yeah, getting back to that, how many new species of these spherophosids, these tarantulas, do you think have been described this year? It is actually a bit of a trick question because it depends on what you view as being discovered. New tarantulas are entering the hobby every month. A large portion of them is undescribed species. These could be unknown to science, or they could also just be quite hard to identify. In the literature, though, we've had at least 4 novel species officially described since 2025. We're going to go through a few of them. So the first is Selenobrachys ostromsupacius. This was discovered on Romblon Island in the Philippines and was described using integrative taxonomy. So that's combining morphology and DNA analysis. This type of integrative approach is going to be foundational to describing new species moving forward and reclassifying older species. At least from my opinion, I think it's going to be absolutely what needs to happen moving forward. Especially when you're considering things like cryptic species, which are genetically distinct but morphologically identical. So the DNA is different, but they look the same. You can also, though, on the other side of that coin, you can get animals that are the same species, but look almost entirely different. You think any example with Let's look at domestic dogs, Canis lupus familiaris. These are all the same species and the same subspecies, but if you were to tell an alien who had just visited Earth for the day, if you showed them a pug and said, oh, look at it next to this Great Dane, these are different species. That alien would probably agree with it. Yeah, that's pretty obvious. They look nothing alike it. But genetically, very, very identical. So it can be really hard to say whether things are what we think they are, or we could be selling something as being a certain species when actually it's quite distinct. And a lot of that's actually going to come down to what species concept you use. Do you use the biological species concept, which tends to be the most popular? So that's whether something can produce viable offspring. But that has a few drawbacks because, for one, what is a viable offspring? Is it just fertility? What about animals that don't reproduce through sex? Are they less viable? Are they more viable? What about an animal that doesn't reach maturity, so it's actually never able to reproduce? What about one that reaches maturity but just doesn't reproduce? There's a few drawbacks in that. One of the species concepts I quite like for the hobby is the ecological species niche. So if you've not heard of that one, that's where we identify animals based on their ecological niche, their habitat. So instead of saying, oh, this is let's say, Psalmopoeus reduncus, so this is the Costa Rican orange mouth, we would maybe say, oh, this is a cloud forest arboreal tarantula. One of the reasons that's quite good in the hobby is that from that ecological niche description, we might not know what species it is, but we know how to look after it. We know it lives in cloud forests, we know it's an arboreal tarantula, so we know the kind of conditions that we need to supply for it. Where that maybe has a drawback is where you're looking to breed these tarantulas, so you do really need to know the species for that. And my favourite of the species concepts is the phylogenetic species concept. So that's looking at the genome, looking at the genetic data, and using that to identify how closely related two individuals are, whether they're within the same species, whether within the right group. And that doesn't take into account what the animal looks like, it is just purely on that genome, which I think is the best way to do it. If you combine that with morphological characteristics, you have a pretty surefire way of describing a species. But anyway, so that's the first one, a cool new discovery. The next one is one you might actually be familiar with. That is A. rufus, Aspinicylus rufus. This one's really cool because it is a newly erected genus, Aspinicylus. It is a species from Javan rainforests in Indonesia and was described in a paper in late January. This is actually a tarantula I recently received. It's just up on the shelf behind me, and it is an absolutely stunning spider. But due to it being recently discovered or newly described, it will absolutely be a wild-caught specimen, which is a topic we'll delve into in a future episode. Wild-caught versus captive bred is a huge topic in the hobby, and it is one that requires nuance and understanding to properly explore. There is definitely pros to wild-caught. There is definitely a lot of consequences to wild-caught, and it's just whether you think those consequences outweigh the pros or not. And a lot of that's going to come down to actually how hobbyists behave in terms of breeding. But again, we'll go into that in a future episode. The next one is a really cool one. And again, it's one you might be familiar with because I've seen a few of these popping up at shows and on social media recently. And that is Formingocylus hattie. So this is a second species from Java, and it was described alongside Aspinicylus rufus in the same study in January. Hattie is actually a Malay phrase that roughly translates to be careful, which is awesome. So for those that are not familiar with scientific names, or binomial nomenclature as we call it, it is a system of applying two names to something. The first is the genus, which is a descriptive of a closely related group of things. And the second is the species, which is specific to that thing and any subspecies that belong to it as well. These names will follow Latin grammar rules, but they can encompass many different languages. Most commonly, Latin, Greek, and more recently, Arabic has been involved. What's cool about Hattie is that it represents decolonisation of science in a way. It doesn't reject the native name for an animal in order to impose one that's more rooted in what we might call Western science. But instead, it references the native people of an area as the true describers of that species. It uses the word that they use. A fundamentally important change in scientific principles, in my opinion. If you go into an area where you're not local to, and say you discover an animal that locals already know about and have a name for, is it fair to say you really discovered that animal just because you wrote a fancy paper from a desk 1,000 miles away? I think it is actually quite important to give reference to the native people in the naming of that species. And I think also in your paper where you're describing it, you should be giving reference to them saying, this is one that is already known to this community. I'm not too sure how I feel about that. I think it's really important to reference the native communities that already know about these animals. I don't like it when people just discover something and act as if they're the first people ever to come across it, which has happened a lot in science with a lot of species. And that's not to shit on scientists that have discovered new species. It's just, I think, an important moral consideration in science. When I was looking at the animals that I have found, I went to great depths to make sure that it wasn't something that was already known locally and already had some sort of name for. If we get to the point where we're submitting that for review, that will go to other experts as well, they might be able to inform me of stuff like that, and I would absolutely welcome it if they did. And if they did, I would absolutely make sure to reference those native communities that already know about this. And then to get onto the last of these scientifically described species, let's say, is Heterophile erdosi, which is a species from Nigeria recently added to the genus Heterophile in early 2025. So this shows that genuses are also still expanding and there is plenty left to be discovered out there. Can't quite remember, but I believe this tarantula was named after a mathematician. Erdosi, I'll have to double check that. I think Erdos does also mean forest in Hungarian, perhaps. I was recently teaching a couple of Hungarian kids, and I'm sure they told me Erdos was forest because we teach them in a forest at the zoo. So it might have to do with that as well. I'd have to check. But I think they're from the African highlands, so it might not be. As I say, I'll check that. But it's so important that we continue to describe species and name new species. For one, it kind of furthers our understanding of the world. But most importantly, it can be nearly impossible to protect what you haven't named. If a species hasn't been scientifically described, it's really hard to enforce protective legislation for that species, which then opens the door to mass whale capture and shipment for the black market pet trade, which is massively profitable and quite influential in some areas of the world. You would be absolutely surprised at how much sway criminal enterprises have in the exotic pet trade worldwide. A lot of it through very legal means as well. It's quite worrying and can be really terrifying for conservation. And I think from that, we will move on to a slightly more positive note. So this is some new research that's recently come out, and a team in Australia have been investigating the peptides in spider venom for potential use in cancer therapy. I really want to dive in to venom in tarantulas in a future episode. At the moment, venom is just a toxin that is administered mechanically. So it is an active administration of it. Poison tends to be passive and it needs to be ingested or absorbed in some way for it to take effect. Both venoms and poisons they're both toxins of some variety. So a team in Australia have been investigating the peptides in spider venom for potential use in cancer therapy. And the preliminary findings from what I can see are actually quite encouraging. With a little modification, these peptides are able to effectively target and destroy melanoma cells, ones that have even developed resistance to other modern treatments. It also appears to halt cells from developing resistance to those treatments in the first place. So it's maybe worth explaining what a peptide is actually. Venom tends to consist largely of peptides and proteins. Peptides are small chains of amino acids linked together by peptide bonds, which are small covalent bonds. These chains are smaller than proteins, and we tend to describe them as the building blocks for proteins themselves. Peptides have multiple important roles in biological processes, so things like hormones, signaling molecules, neurotransmitters, muscle growth, and even now medicine. Tarantula venom is primarily made of a mix of these peptides and small proteins, like we were saying, which have evolved over millions of years to target the nervous systems of insects and small animals. But with a little modification, they're able to move through the selectively permeable barrier of cancerous cell membranes and essentially shut them down from the inside out. So for those of you that aren't aware, a selectively permeable barrier is just something that allows certain things through, but doesn't allow other things through. So in cells, many cells have, pretty much all cells have this cell membrane, and it'll let through very small molecules, but larger molecules need energy to be processed through. So it's almost like little holes in the membrane. Peptides are so small that they can just move through these passive transport systems and gain access to the cell very easily. They don't need any energy to be actively transported across that membrane barrier. So being able to just introduce them and then passively move into cells is really efficient for administering them. This is actually an amazing advancement. So melanoma is kind of known for being the most aggressive form of skin cancer that we're aware of anyway. And although there's quite a few effective treatments already in use, resistance to cancer therapy is a growing problem in cancer research. Because these peptides in the venom work in an entirely different way to any of the other treatments we're already using, they can effectively be used alongside or in place of other forms of cancer therapy that may be failing. It's likely that human trials are still a couple of years down the line for what I was reading, but the preliminary work was so promising that there appears to be some talk about some of the researchers already trying to push it forward, calling for more funding from several governing bodies and charities. A lot of this research is going to comes from the University of Queensland and has been featured in several medical and zoological journals. If I remember, I will link some of those journals down in the description for you. But this type of news is exactly what we want to hear in the news. The days of vilifying spiders are quite far from over, but every positive story is a step in the right direction for us. And when we're thinking about spider keeping as a hobby and exotic pet keeping as a hobby, what we need is more positive interactions in that space. It's so easy for people to say, "Oh, I'm scared of spiders," or "kill it with fire." As soon as they see one in their house, they just smush it and throw it in the bin. We need to be showing them that there's another side to these animals, that they're interested in, that they're medically important, that they're scientifically important, and that they are actually valuable as a form of life, which is something I forget. I mean, not that I find people forget to bring into the conservation, the conversation when they're talking about spiders. So really, the more in education that we can kind of push out there, the better it's going to be for these animals. And that can start with hobbyists. We can do things like we can perhaps contact schools. We can take our collections into schools to visit. We can invite nieces and nephews round. We can get our children into the hobby. We can show neighbours some of our animals and show them that these really are some cool life forms and we should be respecting them. But I think with that, we'll see about wrapping up. Hopefully, you've enjoyed this podcast. If you did, I'd love it if you could recommend it to a few friends. You'll have to let me know what you want to hear from these podcasts in the future. I'm still trying to decide what the release schedule is going to be for them, likely monthly or bi weekly, depending on how popular it is. It's really a format I'd love to push into. It lets me spend a little bit of time, a little bit more time talking about the science without having to worry about the editing of videos as much. So, with that, until next time, cheerio and rasta.